Can we use genetic engineering to replace the electrical lights we've grown to rely on? Electrical lighting takes up around 20% of global energy consumption. What if I told you we could get this number down to less than 5%? What if I told you it is possible now with current science in the field of plant genetics and biotechnology? In nature, organisms produce light to communicate. This is called bioluminescence. Thanks to their adaptive changes to their DNA, these stunning organisms are able to produce an enzyme called luciferase and a substrate called luciferin. The enzyme luciferase breaks down and oxidizes the luciferin. This process produces light. Scientists have isolated the genes responsible for producing these two molecules. These specialized genes can be printed into a circular piece of DNA called a plasmid. This plasmid is then introduced to a bacteria called Agrobacterium tumefaciens and cultivated. After the chosen plant node is sterilized, it is placed into a concentrated bacteria solution, and the Agrobacterium can transfer its genetic material. Then the plasmid becomes part of the plant's genome. After the initial infection of the plant node, the plant is transferred to a tissue culture supported by growth hormones and nutrients. These plants can be easily propagated to replace current lighting mechanisms in streetlights, lamps, signage, and among other outdoor lighting. While dated studies may suggest lumids may be quite limited, if transformed with optimal plants using strong promoters and vectors, along with optimizing codons, these plants have the potential to be quite bright. But how can we control the light? Calathea plants, among others, offer a solution, as they are known to exhibit a circadian rhythm. By isolating and selecting for their genes in genetic transformation, we could induce bioluminescence that only occurs at night. This could change the way we see and light our world, all while reducing the demand for electricity and not only minimizing our carbon footprint, but actually reversing it.
Now that's what I call a breakthrough.